Thank you, Aaron. Nice to see everyone. I hope everyone had a happy new year and is excited for an action-packed SFPM 2026 meeting series. I think I know most of you. For those of you who don't know, Jesse Boehm, Alice Saragni is going to be joining us shortly, but we continue to be very excited about the opportunities ahead of us as your co-presidents for this upcoming year. There's going to be a lot of excitement and a lot of action, and hopefully a lot of value provided to all the members of SFPM. Just a couple of notes off the bat. We're actively working to set up the speakers for 2026. The board is trying to identify some of the strongest speakers that would make for a really action-packed seminar series. If any of you have ideas, please reach out to myself, Alice, Keith, Bruce, any other board member, and we'll collate everyone's best ideas and make sure we have the best speakers. We've gone ahead and added the calendar invites to everyone's calendar by popular demand. Hopefully that'll help everyone reserve the time and attend. You'll see that next month we have a great talk from Wesley Tansey from Memorial Sloan Kettering, who's been aggregating the world's SFPM type of data and has some new machine learning tools that he's used to compute off of it. And beyond that, we're continuing to fill in the content. In addition, we're looking for your ideas on people who are not yet involved with SFPM that you recommend we reach out to in the upcoming year. We have the goal of adding at least 100 new members in the next 100 days. We're particularly looking for folks in the data science arena and other arenas that are not fully covered by the current membership of about 400 people. But if you have ideas, please reach out to your colleagues. Again, please have them reach out to the board. We certainly want to expand the membership base both within the US and internationally. We're working on planning a meetup at AACR. It's not finalized yet, but there'll be details to come. So for those attending the meeting in San Diego, there's a very high likelihood we'll be all be getting together, and Keith's going to be buying everyone beers. So stay tuned for that. Just joking. And then finally, there's work that the board is beginning to think about around strategy and branding for the year ahead. There'll be probably opportunities for everyone to contribute. There's also thought about industry and biopharma sponsorships. No final details yet, but if there are organizations that you think would be great sponsors for SFPM for the year or years ahead, please again reach out to myself, Alice, Bruce, Keith, anyone that you know, and we'll work on collateral and an approach to engage those members. Those are all the announcements. Keith, and others, is there anything else we wanted to say before we go into the science of today?
I don't think so. Just anybody engage who has ideas. We're actively accruing, as Jesse said, all ideas from all members and as to how to improve and how to make changes at this point. Thanks.
Yes, and we've been in touch with Dr. Tony Latai. Some folks may have heard of him. We're hopeful we'll be able to get him to attend the event at ACR and also continue to map out ways for Tony in his new role as NCI director to engage the SFPM in the year ahead. Okay, today I said we're going to have great topics and great seminars this year. Hopefully today isn't a letdown, but we drew the short straw and we do a presentation from my group leading off the bat with Dr. Niklas Rindtorff presenting. So I'm just going to give a couple of introductory remarks and then I'll introduce Niklas. Of course, the way I think we all think about the work we're trying to do together is to try to make precision medicine successful. We're all confident that molecular information provides some priors to guide patient care and to develop therapeutic targets, but insufficient priors. And so we within our lab, and I'm sure in all of your labs, are quite hopeful that functional testing of multiple forms together with molecular profiling can really make cancer precision medicine successful. So in our group, formerly at the Broad and now at MIT, we've been trying to think a lot about what are the missing opportunities? What are the ways in which functional genomics could transform precision medicine over the decade ahead? How do we use large compendium of information like the dependency map of many cell lines and organoids? How do we also develop new technologies to perturb heterogeneous patient samples right out of the patient, as you'll talk about today? And how do we bring those ideas together to eventually enhance patient care? And what are all of the roles that we can play both within our lab and in all of your labs in accelerating a field level transition? We in our lab think a lot about a temporal view of cancer modeling. We're not that interested on whether cell lines or organoids or PDXs or slices or this or that is better than any other approach. We are interested in trying to utilize different types of models for different purposes, again, to enhance patient care. There's been a lot of attention on the right-hand side of this diagram with cell lines and organoids, but today, Niklas will give an update on a new technology we've developed to try to grow and perturb patient tissue within the first 5 days ex vivo. And as Niklas will say, we think that's important because that we think will be a time point in which cellular states and biology, hopefully epigenetic states, will be retained. And so that might lead to more faithful predictions. So that's all I wanted to say by means of introduction to our own group. Today, Niklas is going to be giving an update. He's a postdoc or has been a postdoc in our group at MIT. He's now in Germany. So he's joining us from Europe. He's been really focused on imaging-based approaches using high-content microscopy. And this started from work he did in Michael Boutros' lab at the German Cancer Research Center, really elegant work on understanding how to image and ascertain the vulnerabilities of colon organoids. Niklas received an MD from Heidelberg University, and while he was working in our group in Boston, he also got a master in biomedical informatics from Harvard University. I tried to think of something embarrassing to say about Niklas, but I couldn't figure it out. So, Niklas, only a professional introduction for you. Why don't you take it away?
Thank you, Jesse. I hope everybody can hear me and see the slides properly. Today, I'm going to be talking about the work that we've been doing over the last couple of years in Jesse's lab on studying the ex vivo dynamics of malignant ascites and its use for functional precision medicine. And I think to start this talk, it might be most useful to really go back in time when we started this project around 2020. And what we asked ourselves at that time was, what lessons can we learn from some of the very impressive progress that has been happening on the functional precision medicine side in the heme-onc community? And this was the time when the XL1 study results, at least interim, were published. And now we're already at a point where we have even more data points related to the clinical validity of these functional methods. There's an XL2 study that's underway. St. Jude's has developed a clear, great laboratory developed test, and there are many other examples that I'm not mentioning here. And what we ask ourselves is, well, if we go backwards, what was the precondition to even be able to do these kinds of clinical studies in sort of a manageable amount of time and effort? And I think the second factor that we arrived at was, well, it needs to be a cheap enough assay. It needs to be a scalable enough assay. And I think about a year ago, there was a really great talk actually at the SFPM from investigators at St. Jude's that walked through the evolution of their platform from initially microarray, then fax-based, and eventually image-based profiling. But image-based profiling is not the only thing. I think one has to make sure that the cells that one is analyzing are readily available from patients, and there's not a lot of pre-culture that needs to happen. So trying to avoid organoids or any other intermediate step as much as possible, I think was a desiderata for us. And then last but not least, ideally one is able to cryopreserve samples and ship them around the country. And if we then go one step further back and we ask, well, what are the prerequisites to actually be able to handle samples at such a scale? I think it really comes back down to sort of the biological basics. How do we develop culture systems that are reliable, that are able to keep cells, living cancer cells, ex vivo stable as much as possible? And in the case of the hematological malignancy community, there was some very important basic work happening in the early 90s by Campana, who then eventually also worked at St. Jude's and is now in Singapore, where they showed that if you use these MSC feeders, you're able to maintain the viability of leukemic cells, while if you do not expose them to some kind of conditioned medium or feeder, their viability drops very quickly. So with these three factors in mind, we effectively started out and said, well, what are our requirements? And we added one particular additional requirement, which was again also informed by recent work at the time coming out of Peter Sorger's lab, that showed that there was a real risk of understanding viability effects, especially in solid tumors where the growth rates in vitro tend to be quite variable if one only focuses on the final time point of an assay. And so it would be wise to be able to follow a sample over a time course and to actually really try to estimate its growth rate and how a treatment affects the growth rate, effectively the doubling time of a sample ex vivo. So briefly, these were our starting requirements. We wanted to maintain this 384 well liquid handling interface, both for the microscopy and robotics. We wanted to be able to have continuous label-free microscopy to monitor a patient sample. And we wanted to avoid any kind of pre-expansion, sorting, or filtering, or at least I knew it from doing a lot of organoids, given that just has both biological effects, but also just practical clinical effects on the ability to offer this to as many patients as possible. And the source of sample that we arrived at, and those were existing relationships to the work at the Cancer Cell Line Factory, was malignant ascites. Just as a very quick recap, malignant ascites is a clinical presentation that can be seen in effectively every malignant disease of the lower abdomen, but it's very, very, very often seen in ovarian cancer, and there oftentimes also as one of the first manifestations that patients present themselves, but also in gastroesophageal and pancreatic cancer. And the basic mechanism is it's effectively a form of metastasis where these malignant cells eventually move through the peritoneal wall into the abdominal cavity, and with them they take both immune cells, which we'll get to later, and fluid that eventually then builds up as these barrier functions start to wane. Most of these patients that present themselves with malignant ascites, as I already mentioned, they have ovarian cancer, but there's still a significant fraction of patients that do present with malignant ascites and have gastroesophageal disease, so both adenocarcinoma of the stomach and squamous cell carcinoma and adenocarcinoma of the esophagus. And importantly, these patients, they tend to have a fairly bad prognosis with about 6 months of overall survival once diagnosed with malignant ascites. So we thought this was not only an available source of cells, but also a community that really stood to benefit from introducing a functional assay. So with these requirements at hand and this sample source identified, we went ahead and over the course of multiple months developed an experiment that is just in a sketch drawn out here, where we currently collect samples primarily with our partner Sam Klempner at Mass General. And then these samples get processed within a day, ideally, but there can be latency of up to 3 to 4 days. And at times, we really do not do any kind of additional filtering, seek these cells at about 12,000 cells per well, then perform a time course of about 5 days of live cell imaging. Importantly, these plates already are pre-coded with small molecule perturbations and monitor how the cells react to these perturbations over the course of 5 days, where we analyze the individual images and identify the cells using a computer vision model. So here, just a couple more details. I think this is important, just as I think we're all very interested in the different parameters of these kind of assays. I already talked about the seeding. We coat these plates in gel tracks, which is just a reagent we usually use in organoid culture. And we also use a colon organoid medium. However, what we learned over the course of the project was that if we enrich that medium with some of the native fluid that the ascites comes with, we actually can boost the viability of these samples further. And this is sort of obviously a condition effect, and the medium is less chemically defined as a result, but the benefit and viability makes this worth it. We had about 28 treatments in our initial library and 12 replicates each. We used only a single concentration at that time because our primary objective was really just to see, is this an assay that is reproducible, that's robust. We didn't, at that point in time, seek any kind of clinical actionability and dose response relationship modeling. And the microscopy that we did, as I mentioned, were bright field images as the backbone. So we really just collected at 20x magnification across multiple focal planes the transmitted light. And then to annotate, however, the images and the different cell types and whether a cell is viable or not, we had additional fluorescent labels in parts of these wells and monitored them over time just as well. Now, the primary reason why we didn't just use fluorescent labels, I think that's an important part to briefly mention. There are two reasons. One is we observed this early in the project, and we also now see it later in the data, that just simply fluorescently labeling cells, so let's say the cell surface markers to identify the tumor cells, or markers that are related to cell viability, such as apoptosis reagents, tend to interfere with viability. And that effect tends to really destabilize samples to the point where measuring viability can be challenging. And then on the other hand, we're also seeing that most of these reagents are not optimized for continuous imaging, so they bleed relatively quickly. So both the lack of information over a long time horizon and the viability effects were reasons why right from the start we had the requirement of trying to effectively be able to run this whole assay with just bright field images alone, which certainly then represented the computer vision challenges, which I focus a lot of attention on solving. So with this, we have an overview of our cohort. We had about 45 plates from about 35 samples and about a bit more than 20 patients, 25 patients. And the mix of gastric and esophageal cancer patients was relatively even. Most of these patients were diagnosed initially at already fairly late stages. And obviously, by the time that we collected these samples, they were all at stage 4 metastasized disease. What is, I think, important in the context of this conversation is that most of these patients also had progressive disease. They were on treatment, and most of their treatments were given. The last treatment before the sample was collected was given between a time window of 1 week to 2 weeks. The samples, as I mentioned already, were handled usually in a time window from a day to 3 days. Now, this is the kind of data that we collected. So you can see here what is effectively our standard panel, where we have effectively 50% of the wells that we monitored. We monitored not only the bright field information, but we also collected these fluorescent labels over time. And you can see here on the left, the bright field projection. So we have different focal planes to better observe the boundaries of the cells, as you can see here in fake color. And then in the center, we have 4 different fluorescent dyes in a false color image, where we have caspase activity and a mitochondrial dye, PMRM, to contrast both dying and fully viable high mitochondrial membrane potential cells. And then EpCAM as a marker of the epithelial malignant cell compartment, and CD45 as a marker of a very broad contaminating immune cell compartment. We did not observe many additional cell types. Primary additional contaminant were erythrocytes, which were very easy to identify. And then the next step, obviously after collecting these data, was to then manually and semi-manually annotate these images. So you can see such an annotation mask here on the left, where we separated 4 different cellular states: viable, malignant, dead malignant cells, and their immune counterparts. The task, sort of to just jump perhaps just slightly ahead towards the computer vision problem that we are working with here, is called panoptic segmentation. So we don't only want to know which pixels in the image belong to which cell, but we also want to really be able to count cells. And this is obviously important, as this is our primary readout. So we want to know how many cells and what type of cells do we have in our image. We also developed this immune cell panel to get a bit more granularity on the immune compartment that we're working with. And this is work led by a recent master student, Darren Takimian. And using this panel, we're able to further differentiate the cells on the immune compartment to lymphoid-like NK and macrophages. Today, I'm not going to be spending much more time talking about this immune cell work, but this is obviously something that, especially as we're looking into studying immune cell interactions with malignant cells, is of great interest to us. Now, as I already mentioned, the first part of this project was really after we had collected these data to train a computer vision model to detect these different cell types, to classify them. And here you can just see two effective instances of how this looked like in practice. So we have 4 different cell types: viable malignant cells, non-viable malignant cells here in red and green on the left, and then on the blue and yellow we had the non-viable and viable immune cells respectively. And this was in the beginning manual work, and then over time one was able to train a computer vision model to help with the annotation, and it was mostly a review process guided by effectively the fluorescent labels. Now, there were also certainly challenges, and these challenges, they currently still exist in the data. The model is able to still learn robustly, given that we just have annotated a sufficiently large amount of these images, but there certainly is room for further improvement. We, for example, have these crowding artifacts that you can see on the left, where it's just very challenging to sometimes know where the boundaries of a given cancer cell are. In the middle, you can see a late time artifact where on the one hand, the fluorescent dye intensity starts to diminish. So the ability to identify different cells drops. And we also have some contaminant overgrowth of fibroblasts. And then on the left-hand side, these are just very basic mechanic artifacts when we see an uneven illumination in the plate. Now, despite some of these difficulties, we collected a large data set of about 500 annotations, 50,000 annotated cells across multiple patients and plates. And these are just some examples that showcase just how heterogeneous these samples can be, where on the left-hand side, we have samples with a very high fraction of viable malignant cells, while on the right-hand side, we have samples where there's effectively no viable malignant cells. So in these cases, it's obviously impossible to very challenging to be able to measure any kind of treatment effects on the viable malignant cell compartment. This gives us sort of the full overview of the core that we created. So I already introduced you to some of the clinical metadata. Now we can take a look at the kind of data that we have on the imaging side for each of these samples. So we have usually about at the order of 10,000 to 40,000 fields of view imaged across different channels. We follow most of these samples for a time between 5 and 7 days. And as I already mentioned, we usually annotate about 10 to 20 images per patient, which gives us overall a data set of about 500 annotated measurements. And I think this really brings back brings home the dimension of this data set, which we call the ascites predict 1 data set. So we have about 18 million images that we acquired overall over the course of multiple years since 2022, which represent about 120 terabytes of data, which we can obviously compress further. And I think at this point, technology has really caught up so much that we are pretty optimistic that we can share most of these data upon publication. 1 million fields of view, 90,000 trajectories, so effectively movies of cells. And as I mentioned, about 50,000 annotated cells with these labels. And this is how, once the model is trained, and I'm going to go a bit more into details of how the model is trained, the data analysis results look like. So we have this effectively choppy video stream of a well over time. Here we see about 72 hours of trajectory, and then we have the one-to-one mapping into our 4 states. Now, obviously, what's most interesting is to look at treatment effects. So here we can, for example, see a positive control, and we see that most of the malignant cells instantly flip into the STAT state as they react to the proteasomal inhibition. And the way that the model works under the hood, I'm going to touch it only just very briefly, but I think it's important to give everyone here an intuition, because I think this technology is really here to stay and hopefully can serve many other projects that are going to come about in the SFPM, is based on a model that's called CellPose. This is work coming out of the Genelia campus of HHMI. And the core idea is that we want to train a model that is able to segment cells. So we identify the boundaries of every given cell, but we also want to classify the state of each of these cells. And the first problem was already solved by the Genelia team using this approach, which is called a central flow. And the basic idea is that we annotate a cell by really demarcating its boundary in a high fidelity way. And then from this boundary, we can derive what is called a flow towards the center. And we teach a model to learn this gradient from the boundaries towards the center of the cell. And this leads to very high quality segmentation results that you can, for example, see in this slide. The loss of the model is then both this flow so that we can segment cells and the label. So what kind of cell we're looking at. And it was this addition to also classify the cells that was the individual extra step that we made where we fine-tuned such a model both to these new cell types and to also classify them. The model has about 300 million parameters. We trained them for 200 epochs, which is fairly standard in the imaging community. And we use this existing model that we just fine-tuned effectively for this purpose. Now, the model itself has a relatively high fidelity. I'm going to show some examples of this. But I think what might be interesting for this community here is not only whether such a model works, but also how much data is required to train such a model. And what you can see here are some metrics that represent the performance of a model. So as I mentioned before, the task that we're solving is we want to both segment cells. So we have one aspect of quality that's called the segmentation quality. So can we actually see the boundaries of the cells? And then on the other hand, we also want to classify each individual cell that we're observing, which is called, in this particular context, recognition. So how good are we able to actually assign a given cell that we've segmented to one of the four states we've described? And what we can see is that these pre-trained models, they come out of the box already with a very good segmentation quality. This is the central plot that you can see, whereas even if we just have a handful of annotated images, let's say 4 annotated images, the performance is already very high and it doesn't necessarily improve as we add more training data. So all the way up to multiple hundreds of annotated images, while the recognition quality, this classification, which is a capability that we added to this model, definitely improves as we add more data and eventually only plateaus at the order of hundreds of measurements. And what this means, I think, for everybody in the audience, if you go and you want to pursue using some of these more advanced segmentation and classification methods, you can be, I think, at this point in time, be fairly optimistic that a model that's been pre-trained and is available to you is already going to be very capable in segmenting the cells in your image, but it will probably take some additional work, which luckily is way easier when the segmentation task is already solved, to then classify training data and train the model to also be able to classify these different groups properly. Now, how good does this model work? I think there are many ways in which you can estimate this, but I think one way that might be very intuitive for people that know how to pipette is to use a cancer cell line and to seed it into plates where we know how many cancer cells to expect that are viable, and then to use this vision model to predict the number of viable tumor cells within a given image and see whether these two line up. And in this particular case, I think this is the case, and we can even see this really from hundreds of cancer cells per field of view to what you can see here is effectively less than 1 cell per field of view, with 10 to the 0th power. The way that this plot is able to show the sort of less than 1 cancer cell per field of view is because per well, we're acquiring more than 1 field of view. So if there's just 1 cancer cell in the well that's visible, we're going to pick it up across one of the 6 different fields of views that we have. I saw that there's some messages in the chat, perhaps just because some of these aspects were fairly dense. Jesse, if you want to read some, if they're related to the
There aren't
talk,
any
question.
there aren't any scientific questions, but yes, if people have any questions, they could either put them in the chat or just raise their hand or ask them.
Awesome. Okay. So with such a model at hand, we can then go and we can actually count cells. And this is, again, the video, but now rolled out, where we see that the number of malignant non-viable cells is increasing under treatment with bortezomib. Well, this is not the case, obviously, with DMSO. So now, how do we go from these counts to some kind of statistic that we can use to try to get, hopefully eventually guide therapeutic decision making in the clinical context? And I think the first step that one has to do is to aggregate them into these count trajectories. So here, please pardon that the color legend is slightly changed. In this particular case, we again have our 4 different cell populations, and we can say that, and in black, we have just the number of total cells over time. And we can see that as we add bortezomib to a plate, the number of viable immune cells and viable tumor cells here in green and blue drops very quickly, while the number of dead tumor cells and dead immune cells sort of in lockstep also increases and eventually plateaus. In this particular example, the total number of cells is also fairly preserved, which is, I think, a fairly good indicator that the model is also able to effectively maintain an overall count of the cells within a given sample. So once we have these trajectories, these count trajectories, we can obviously measure them for all kinds of treatments in our plate. And then eventually we want to fit a statistical model to go from these trajectories to a number that we can communicate. And the statistical model that we use here is a model that's already been known and used in vitro cancer cell line biology for quite some time. It's called the Gompertz model. And effectively, the Gompertz model is a modified exponential growth model. So we're all familiar with exponential growth, a cell line that's doubling at a fixed time interval or a given rate would eventually have an infinite amount of cells in our dish if we kept it for long enough. That's obviously not very realistic. So we introduce a third parameter, this parameter B, that effectively dampens the growth rate over time as nutrients get consumed. And the final end result is this kind of growth trajectory where with a given starting cell number N0 and a given growth rate A and this dampening parameter B, we can describe how the cells proliferate or also die over time. If a cell population decays over time, this parameter A is negative. And if the population grows over time, this parameter A is positive. Now, something that I'm not going to go into much detail on today, but that's something that I think we in the group are pretty excited about, is that the statistical model that we're using to fit this likelihood to our data can be made fairly complex. And we're taking a hierarchical Bayesian approach where we're able to borrow information from neighboring samples, but also introduce known priors from clinical genetics. For example, we also introduce whether a sample is HER2 mutated, or we can at least do so. And this certainly improves the ability of the model to capture the fidelity, to capture the dynamics more accurately. And I think it might be a bit of a pointer towards the direction that at least excites me about functional precision medicine, where over time, hopefully, we do not tend to think about functional precision medicine and these functional assays on the one hand and genetics as these very dichotomous fields, but really as one integrated mission to use genetics as a prior and then use these functional results to update the prior to get a more refined overview about the kind of therapeutics that we would like to recommend to a patient. So these are some examples of different patient samples and the fits that the model generates to the data. So here at the top, we can see the count of viable malignant cells. And we can see that in certain cases, the count of malignant cells increases over time, in others it decreases. While in the viable immune compartments, the immune cells under the conditions that we've chosen were never able to to stay stable, they always decayed in viability over time. I will later go into some of the details, some of the factors that we found that explain the variability in this starting number of tumor cells and whether some tumor samples grew and why others didn't. But for now, I'm going to spend a bit more time and keep talking about how do we go from such a learned model to an actual number that we can communicate. So when we fit this model, the kind of resulting one resulting parameter that we observe is this factor A that I already mentioned. Just as a quick recap, this is this growth rate. And for every sample, and if for a given condition, let's say DMSO here, we're fitting the statistical model and we're observing a growth rate both for the malignant cells here in purple and the immune cells here in orange. And the general trend that we observe is that the immune cells are more sensitive to the culture conditions that we chose, and so they tend to decay quicker. And you can see that effectively all the immune cells here in orange, they tend to have a growth rate that's negative. They tend to spontaneously decay. I also mentioned that some malignant cells tend to proliferate under our culture conditions, and you can see that in the case where the growth rate is larger than 0, obviously. But in most cases, immune cells tend to be more sensitive to the conditions than our malignant cells. Now, if we add a treatment such as tucatinib at about 2.5 micromolar, we again see a fairly similar relationship where again immune cells tend to be more sensitive in most cases. Now, what we want to do is we want to somehow reference these two, and a very simple way is to subtract these growth rates and arrive at something that we call delta A. And delta A is effectively the difference between DMSO and tucatinib. And this gives us now a fairly good overview about in which cases the immune cells are more sensitive to the drug versus cases in which the malignant cell is more sensitive to this particular treatment. In this particular case, I think we can see one outlier or fairly strong signal where there's a malignant cell that is fairly sensitive to the treatment, while the immune cells tend to experience no effect relative to the DMSO treatment. And this is actually the only HER2 positive cell in this particular plot. Now, if you look a bit closer, you will also see that there are some cases where you see a similar relationship, but at a higher growth rate, obviously. Now, one next question, once we have this metric, is, well, how sensitive is this metric and what are the primary determinants of sensitivity? And what I mean by sensitivity is, can we pick up a given treatment effect? And what we're oftentimes, or effectively in every context, able to detect the effect of a toxic compound at high concentrations, such as bortezomib, we can only detect treatment effects such as of this HER2 inhibitor, tucatinib, obviously when we have a relatively large number of tumor cells. And you can see here on the x-axis a relationship between the number of tumor cells on a log plot and then what we call critical difference. So effectively, if we take the confidence intervals in this Bayesian model, so the credible interval, and we calculate what is the kind of difference, the kind of treatment effect that we can only observe given the uncertainty that we have about our parameter, we see this clear relationship between the number of tumor cells and the critical difference, which I think makes intuitive sense. You would feel more comfortable about an estimate of a growth rate effect if you had hundreds of viable tumor cells that you perturb versus 10. And very, I think, analogously to this intuition, we recover this relationship here. I think what this shows us, however, is that already with relatively few tumor cells, so at the order of 10 tumor cells per field of view and perhaps even lower, we're able to detect treatment effects for compounds, and especially if the treatment effect is very distinct and very strong, such as the kind of treatment effect that we would like to see for a target therapeutic, we feel fairly confident that even with single-digit viable tumor cells per field of view, we can recover these kind of outlier responses with relatively high certainty. Now, I already mentioned that in order to move such an assay into the clinic, we obviously need to know also what are the factors that can be a source of nuisance, a source of issues with the assay. And I think we can really go through some of these nuisance factors if we just stick to the statistical model that I defined previously. So I mentioned that one aspect of this model is the starting number of cells. So we can ask the question, what factors drive the starting number of cells that we have? And one relationship that we found that's relatively strong is that the longer the sample is sitting at room temperature, or even if it's cooled down to 4 degrees Celsius, the lower the number of starting cells is that we observe. And this relationship is seen already even after 24 hours of waiting time. Now, we can take this one step further and develop some form of guidelines for the laboratory where we combine this insight also with additional characteristics that we've learned of these samples, where, for example, given that tumor cells have a larger diameter than most immune cells, the average diameter of the cells is also a pretty good proxy for the tumor cellularity. And with these two in combination, we can find these guidelines for the lab where we say, a sample should be processed within 24 to 48 hours. And ideally, we would like to have average diameters of 12 micrometers or larger. Now, the second parameter that I haven't spent much time on is this growth rate. So why do certain samples proliferate over time while others do not? And one aspect that I only mentioned in the beginning, but I haven't yet shown any data for, is staining. And what we're seeing is that certain dyes have an effect that's equivalent to CDK inhibitors on the viability. And this is especially TMRM, which is a mitochondrial small molecule dye. And others do still have, although not as distinct, do still have a noticeable viability effect. For example, EpCAM antibodies that target the cancer cells, and we can see macrophage engagement using this EpCAM antibody, which I think opens up some direction to think about staining reagents that don't have FC termini, but generally we just try to avoid staining reagents overall. And a second aspect that we identified, although this association is fairly weak, given that we've only collected these data for a few samples, is that we can predict the growth rate in vitro from the mitotic activity of the sample even before it has been seeded. So if we take a biopsy, and these are actually cryopreserved samples, so even if we cryopreserve the sample in the meantime, and we run a single-cell RNA-seq and look at the cell cycle phase distributions, we can find some associations where having a higher G2M fraction does correlate with a higher growth rate in vitro in our assay. Now, when we look at the different treatments that we've tested, we see generally a very wide range where once we subtract the effects of the treatment versus the MFO and of the immune cells versus the malignant cells, the spread of the values is fairly constrained. What's most interesting is not necessarily these marginal effects, but really going into the individual cases and trying to observe whether we can recover some clinical genetic associations. And this is, for example, in the case of HER2, the case where we have here on the x-axis, we have again, effectively our statistical power, the number of cells per field that we're observing. And on the y-axis, we have whether a treatment is detrimental to the tumor cells, if it's a negative delta A, or non-detrimental if it's positive or 0. And we can see here the HER2 positive samples that tend to be on the margin detrimental to the viability if they're treated with tucatinib, while the overall viability effect is more moderate. Now, I think one anecdote that I would like to share in this particular context is you can see here on the left-hand side, you can see a HER2 positive sample that has very little number of cells, so less than one cancer cell per field of view. So very, very scarce number of cells. And here the model, however, is able to build some confidence in a detrimental effect, which is in this case guided also by the clinical genetics prior that goes into the model. So here, I think this might be one example where the model has some prior expectation that tucatinib interacts with HER2, and then because we only have so few cancer cells in our data, it's not able to update that prior much, but is still able to, in this particular case, give the recommendation, if you wanted to say it this way, that tucatinib would be a wise choice, just given the genetics, and it's not able to update. Well, here, if you look at this other HER2 positive case in the center that tends to show that there is no effect of tucatinib, this also had the same prior of tucatinib most likely being active, but here there was sufficient data to update that prior towards saying that, effectively saying that this treatment would probably not be particularly effective for this sample. Now, I think at this point, I hope I've given you a fairly good overview of both the laboratory process and the statistical analysis process that hopefully gets us to the point where we can make statements about the efficacy of a small molecule in the clinical context one day. But I think the most important question that we wanted to answer when we started out was, how reproducible is this? If we take samples, 2 samples from the same patient, do these predicted delta A values, do they correlate? And the answer is they mostly do. So here what you see is a bootstrap where we take effectively off the 40 or so plates that we've measured, we take these delta A values, and we just calculate the correlation across all of these different plates. And we compare what is the distribution of correlations between randomly picked pairs or pairs coming from the same patient. And we can see that highly significantly, samples coming from the same patient tend to show strong correlations of these delta A values. And this is rather anecdotally and needs to be further investigated. In those cases where we do not see this correlation, actually the primary source of that lack of correlation are logistical artifacts where once the sample was collected the very same day and one plate was started right when the sample arrived, and another might have been processed only a couple of days later, which then actually tends to be fairly detrimental to the sample. So I hope with that I was able to show you in summary that we can maintain malignant ascites samples from gastroesophageal cancer patients a sufficiently long ex vivo to perturb them directly. We can use label-free microscopy paired with fairly modern computer vision methods to sufficiently detect, count, and then model treatment effects on the malignant cell compartment, and also identify some already known genetic associations. And then finally, we can see in these retrospective analyses some factors that influence the sample quality and accuracy of this kind of modeling approach. And with that, I am about to close. I think there obviously are many, many further directions for this kind of work that we're excited about. Primarily, obviously, go towards dose response relationships, which is already ongoing, and we have collected the first number of about 10 plates. Also investigate the way that we can measure immunotherapy activity, and there's one direction that I think is very promising in the context of using imaging, namely that we can also measure cell-cell interactions and how the count of cell-cell interactions and the different immune cell types changes over time. And then finally, let's try to take this into a more clinical context and also observe systematically the patient's treatment response and see whether we can find some correlations. And with that, I want to thank you, and I want to thank my colleagues, Jesse, Samuel Kuntner, and Mushrik especially. And Mushrik is right now in charge of high throughput science at the Koch Institute, is really doing most of the wet lab infrastructure at this point. Samuel Kuntner is our partner at MGH, and Jesse is keeping it all together. Thank you. Happy to take any questions.
Thanks, Niklas. Let's take some questions. We have about 10 minutes.
I had a question about, so you showed the nice correlation with the ER positive, and then there were some that were ER negative that had the reverse. Sometimes these, and you had the one ER positive that didn't show response here. Did you dig into those and find any interesting validation, like there was a mutation in the estrogen receptor, or or HER2 or something that block the agent from working or anything like that kind of was interesting.
It's a really good question. We haven't done that yet, but we really should consider it. Especially that I think one important aspect or caveat in this particular context is that the time point where the HER2 status was determined by the clinical geneticist or the pathology lab has quite a bit of offset temporarily from the time point where we collected that sample. So in some cases, if you would look and you would sequence, you might actually see that the tumor genetics have developed quite a bit.
Yeah.
Yeah, Niklas, I don't know if it was this one sample, but there was one particular sample where the diagnostic sample is HER2 positive, and then the single cell RNA-seq at the time of the sample had shown clear downregulation of HER2, and that could explain situations like these.
Yeah, just my experience has been the functional is usually quite accurate and quite revealing for the genetic associations where it's well established, and it usually finds errors or resistance mechanisms quite well on anecdotal basis. So usually those are good points for discussion in a paper.
Absolutely. Well, pathologists never get it wrong, Keith. So,
yeah,
functional tests.
I don't know about that, but lawyers say otherwise.
Why don't we go to June and then Christopher? June.
Great talk. Sorry, I had to step away in the middle, so I probably missed some of the details. Two questions, both of which are a little bit technical. One is I see that you have an antibody panel that you're using to stain immune cells, which I think will be very, very valuable. Can you remind me, do you have to wash, especially for suspension, like, leukemia, that sort of thing, it gets very, very difficult to do.
Yeah, so we don't wash. This is a very crude protocol. We effectively add these staining reagents to the culture medium at the time that we plate these cells. And this obviously then creates all the negative toxicity effects that I described. Right? So it's a double-edged sword, but there is no additional washing or liquid handling involved. This is all live. And once we have the cells plated, we do not interact with the sample anymore.
And then June, there's bright field classifier development for each of the immune states, which we're in the middle of. And so once you're confident that the bright field can predict the states, then you can get out of the business of having to do the stains.
That would be great. I mean, especially for people like us doing leukemia sensitivity, drug sensitivity testing. It's just such a pain to stain them. Second question related to that, we do a lot of leukemia stuff, and for most of the immune cells, they have a huge nuclei, and it's the feature capture is a lot more difficult compared to solid tumor. I mean, I've seen that it seems like you're doing both. I wonder if you can comment on that and maybe some caveats of the imaging analysis in the context of hematologic malignancies.
So I need to make sure I fully understand the question. Maybe I clarify first one thing, which is that we do not actually stain any nuclei here. So this is one important caveat. We do certainly see serious nuclei size differences between the epithelial cells and the immune cells, which I would reckon is harder to do if you have a hem-onc sample and you compare that to the environmental immune cells. So here, because we're not directly staining it, I can't give you an example of the overall dimensions, but in some instances it's pretty visible and the size difference is considerable. Now, maybe with that information, can you try to rephrase the question, or maybe I already answered what you were asking?
Well, for example, you're using other dyes, mRNA, look at things like mitochondria, et cetera, because the nuclei for the leukemia cells are so huge, it's very difficult to see other organelles compared to, say, solid tumor cells, et cetera. I just wonder whether that creates a problem for the downstream analysis.
I see. I would say rather no. Perhaps TMRM is not a really good dye for immune cells. So, for example, in this image, you can see that macrophages, especially activated macrophages, they do tend to have noticeable abundance of mitochondria that also stains well, while especially lymphocytes do not tend to show them. The reason why I'm still not yet pessimistic about generally these kind of staining approaches is that, for example, this caspase reagent, which locates to the nucleus, is fairly potent in identifying apoptotic cells, and that locates to the nucleus. I would even almost dare to invert and say that probably the computer vision problem might be slightly more tractable with hematological cells, given that they have these more regular shapes. But I would love to take a look at some of these images and see how it works. So if that's something you can share, that'd be very exciting.
Why don't we keep going? But maybe June, we can connect you with Daron, the lead on the immune side of things, and you guys can compare notes. Christopher?
Yeah. First of all, beautiful presentation. I thought that was really innovative work, very, very well presented. I had a question regarding the dissociation of cells for the assay. That happens a lot in ovarian malignant ascites. The cells are actually in these large aggregates. And I was wondering, to what extent have you had to dissociate the cells, and how you're thinking about that impacting the sensitivity of the cells in this in vitro assay to the different chemotherapies that you're applying. And apologies if I missed that part somewhere during your presentation.
So we were mostly not disaggregating the cells. There were some instances where we had rather clumpy samples, but on the margin, these are fairly isolated samples. We do some erythrocyte lysis if the sample is very bloody, but we do not do any kind of additional digestion in most cases, as far as these I am aware of. This is certainly different once we would go into different malignancies where the cells tend to be more adherent to each other.
Yeah, and maybe that's something that you could approach with some kind of like physical dissociation. I don't know how that would impact the cellular sensitivities to different therapies, but I think it is a solvable problem. I was just kind of curious about how you were approaching it. And one other thing, I really love the approach that you're doing, this time-lapse microscopy. Are you seeing differences in the rates of cell death being induced by different types of agents in that time-lapse microscopy that's then showing you differences between classes of drugs? And then how are you incorporating that into your thinking about how to eventually score what is toxic versus what is not toxic?
Yeah, so this statistical model is fairly crude. It has only effectively one parameter that we're fitting to each treatment, which is this rate. So by definition, that means the rate is always constant over time, and it just gets moderated by this one global parameter that's learned for all treatments. So to answer that question, the answer would be right now I'm effectively avoiding to look at different temporal effects. That said, I do see some differences, and I think right now, however, those differences are most likely artifacts because some concentrations that we chose were so potent that they just instantly degrade viability. Looking forward, one thing I'm really excited about is not necessarily perhaps the dynamics and whether there are differences in dynamics of cell count, but differences in dynamics in cell-cell interactions, which I think might be really interesting for more immunotherapeutics. So this is something one can look at.
Christopher, I think we need to wrap up, but I think we're trying to look for really strong outlier effects that might be refractory to some of these things. We're kind of less interested in very small effects. So I'm hopeful that the very, very strong things will be seen regardless. Related to the first question, we're trying to do this with sarcomas and other malignancies as well, where you might have to disaggregate samples. We don't yet know whether the mechanical or enzymatic digestion conditions, if you move beyond ascites, for instance, to tumors and chop them up. Colin's trying to figure that out, but we suspect some of those variables will be important, others won't be. But we'd love to collaborate with folks on this one. I think we need to end the session for the moment, but it was great to see everyone. Niklas, great talk. And if there's additional questions, please send them our way. We'll see everyone next month for the seminar series led by Wes Tansey, and please be in touch on any of the topics I led off with in the beginning. Have a great day, everyone.